আইএএসডি করছে জীবন কেটে, আইএএসডি করছে জীবন কেটে বেকারত্বের জ্বালা, চাকরি জীবনের কাটতে রিবন, চলে এসো এই বেলা, চলে এসো এই বেলা। দিনকাল একদম ভালো না, চাকরি আজ খুবই রেয়ার, প্লেসমেন্টের দায়ভার আমরা তাই করছি শেয়ার। আইএএসডির হাত ধরে হবে তোমার প্রথম রিক্রুট, তাই সব টেনশন যাও ভুলে জীবন এবার হবে রিবুট। আইএএসডি করছে জীবন কেটে বেকারত্বের জ্বালা, চাকরি জীবনের কাটতে রিবন, চলে এসো এই বেলা, চলে এসো এই বেলা। ইংরেজি টেকনোলজি ভয় পাও বুঝি, হবে এবার সোজা, যাবে কোথা খুঁজছো বুঝি, এসো এখানে শেষ হবে তোমার খোঁজা। আইএএসডি করছে জীবন কেটে বেকারত্বের জ্বালা, চাকরি জীবনের কাটতে রিবন, চলে এসো এই বেলা, চলে এসো এই বেলা। জব গ্যারান্টি সার্টিফিকেট সবই চাও বুঝি, ভুলে টেনশনের বোঝা, আইএএসডি ঠিকানা তবে এখানেই শেষ পথ খোঁজা। আইএএসডি
एहसान है मेरी सांसों पर तुम्हारा, अनगिनत एहसान है, तेरी खुशहाली रहे कहता मेरा ईमान है। मेरी सांसों में तिरंगे की बुलंदी की दुआ, मेरी सांसों में तिरंगे की बुलंदी की दुआ। मेरे हिंदुस्तान तुझ पर जान भी कुर्बान है, मेरे हिंदुस्तान तुझ पर जान भी कुर्बान है।
Hello everyone. Good afternoon, Tawfiq. Good afternoon, Tawfiq, Shantani. Hi, Shahana. How are you all doing? Great to see you. Hi, Priyanka. Quickly join everybody. We'll start today's session. Um, give me a moment. You guys quickly join in. We'll start soon. Hi, Rishi, Basudev, everybody, welcome. I'm doing good. Thank you. How are you doing, Tawfiq? And congratulations. I saw you have published in an research paper. So many congratulations to you. And I'm doing great, thank you. I'm still with my morning coffee. It's early morning here, and today there was supposed to be sun, but so far no sign of sun. So I don't know how good or bad a sun date will be. Hoping for best. We were planning for a little bit of barbecue party after our sessions today. So I'm hoping it becomes a sunny day. So far, there is no sign of sun. So in London, if it is a sunny day, it's very good. Otherwise, sometimes, as everybody knows, London has a bad reputation of being grainy and gray. So some days it is absolutely gray, and then it becomes very, gloomy, and you don't feel energetic. But then when the sun comes up, it's really, it's really nice. You feel energetic, you feel nice. Yeah, and this is summertime, so summer is really nice here. We have longer days. You can go around, people go crazy, they're like out in the weather, going doing picnics and all that stuff. uh, okay, Rishi is saying same here in Birbhum also, ma'am. Kalbushaki is hitting hard. Yeah, this is the time for Kalbushaki. So did you guys get any mangoes after Kalboishaki? Just okay. I see nine people. Give me one moment. I'll just quickly start. And thank you, Yasmin. Good afternoon. Thank you for joining, and a warm welcome to everyone. Today there is no quiz, but because yes, last day it was a very small, like we had a few concept discussed, but then today we will cover a little bit more in depth, two three concepts, and then about the clinical data management role, what do they do, what a daily life looks like, what is the purpose, like why we do what we do, what is the overall layout of a responsibility of a clinical data manager, all these things. Yeah, that's true, Rishi. That happens to my mama bari as well. It's like everything fell down last round of Kalboishaki, and then it's only for pickle pickle everywhere. So true. So yeah, so we will discuss about the roles and what the roles and responsibility that a CDM does, how a daily life look like, and then what are the roles once you get into the CDM role. Then what are the hierarchies, how you can progress in your career through the roles, what are the roles available um, within CDM, so how you will progress on the career ladder. We will discuss that also a little bit, and then how the role changes. This is the overview of for today's class. I'll just start in a minute. Yes, we have 10 people. We'll start the class today. I'll just share my screen. I hope everything is visible. Yeah. So let's begin. So just a quick recap. How it goes. If anybody new joining in, please do consider to introduce yourself here. And any question, any comments, anything that you want to know, keep putting on the comment box. Will time and time we will review and discuss them. Okay, so just a quick recap of what we learned the last class. So last class we learned about few basic concepts of clinical trial like informed consent. We learned about protocol, cohort, what is placebo, what is investigational product, and then we also discussed about control groups, endpoints, and interim analysis. And if I remember, we also discussed a little bit about randomization and blinding. But randomization and blinding is a very fundamental uh, concept for clinical research or clinical trial. So we will discuss that in a little bit detail today to understand why it is done, how it is done, what is the utility of it. Okay. So randomization. So okay, Monish Dandapat. Hi, Monish. Good afternoon. I think I'm seeing you new in the class. If you are, then please introduce yourself. Where are you from? What are you pursuing? And welcome to the class. So, what happens? Randomization. So, clinical trial, why we do it? We want to know, we want to test the effect of a drug in a wide range of people. Okay, so we cannot we have to really expand our horizon and test, um, even though it is a very limited area, because once the drug is in the out in the market, there will be plenty of other people who will take. And in respect to when it is out in the market, what we are doing in the clinical trial is still a very less number of people, less number of population that we are testing the drug. But then also, in clinical trial process we have to replicate the diversity. So we have to include all the age, like different ages, all different communities, location, and then age, race, so we have to have variety so that we can say that the drug which is finally out in the market can if be effective on every people. Okay, so let me give you a context. So medicines when we take, if you see the back of the leaflet, so if you see the label, so I would request you from now onwards, whenever you see a medicine strip, you go and check the back of the strip and check the label. So there, be it medications, be it any cosmetics, there will be a storage recommendation. Okay, so keep it in a cool place, keep it in a dry place, don't bring it on the direct sunlight. So why all these things are there? Manish, hello, ma'am. Myself, Manish Dandapat. I'm from Mednipur, West Bengal. I have completed my B Farm degree and I'm searching for job opportunities. Thank you. Okay. Welcome, Manish. So what? So the store, why do you think the storage instructions are there? So what happens, whatever medicines or whatever drugs that we are using, whatever components that we are using in the drug, uh, in the formulation, that needs to be stored in a particular um, environment, so that its efficacy remains intact. If we disturb the storage condition, that it might affect the effectiveness, and the quality, it might get damaged also. So depending upon that, we have to check in different climates. So many drugs, there will be many drugs which can work in these kind of areas where there is a colder area, okay, but then that might not work in the hot area, like hotter climatic area, because of the storage conditions. So how do you know that where it is working, where it is not? So we have to have a wide variety of geographical location also, and the group also. So to test all this, you know to test the drug in the variety of people, we need to uh, run our trial biasless. Bhashar Nashkar. Hi, Bhashar. Welcome to the class. So, randomization in clinical trial is a process of assigning participants in different groups. Okay. So, previous day we discussed that we have a control group and also we have a treatment group, right? So, how do you decide that who will be going to the control group and who will be going to the treatment group? So the clinical trial, we through the clinical trial, we want to achieve some information which is away from bias and through integrity. Okay, so I'll tell you a funny fact that many of you would except that, suppose somebody is having some, maybe you have hurt your leg and then it's hurting, or any ailment that you're having, and then once on you are on your own, you feel very, it's painful, it's distressful, but then once you go to a doctor, and then doctor says no, 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 it's not much, you take rest and you take few medicines and you will be fine. So many a times what we say that half disease is cured once you visit the doctor. So uh, when you are ill, it is physical, definitely, but to some extent it is psychological also. Not in real sense, but you feel that okay, doctor said and gave me reassurance that okay, this is not much, then I'm feeling a lot relieved, or maybe I'm having a little less pain. So there is a lot of psychological aspect associated with it also. So same happens when a drug, suppose, is given to a patient, and then if I know that I'm having stomach pain and I had a painkiller, it is definitely because of the drug, it is symptomatically relieving my pain, but also giving me psychological relief that okay, I know that I have taken a medicine, and then very soon after the medicine you will feel okay, now my pain is reducing, even though it might take 1 hour for the absorption and to action, it should start acting. But then immediately after taking the medicine, because now you know that you have taken the measure and it will help you reduce your symptom. So there is a lot of psychological aspect that is associated with the treatment, right? So it is a wide saying that once you visit the doctor, half of your ailment is gone, in general, if not so the doctors also, they treat, if you are, if he or she is a good doctor, they will you know deal with you sympathetically, with they will talk to you nicely and give you reassurance that nothing happened, be patient, everything will be fine. So there is a big psychological aspect is associated with your treatment. Now, once we know that there is a painkiller and it reduces pain, then you can afford to associate with your psychological feelings. But when you are trying to prove whether it is reducing your pain or not, that time you cannot have bias, right? That time, first of all, you should you should maintain the integrity of the trial, and it should be away from bias. That because there are a lot of trials going on which is based on the pain relief, and then psychotic drug also, and then other drugs also. But then if you know that you have taken the right medicine, and then if you feel that yes, my because I have taken the drug, I might be feeling better, then it might hinder the result of the clinical trial. So that's the reason we have to keep it blinded. So today we will discuss randomization and blinding. Randomization and blinding goes hand in hand. So it will be we will discuss it, it will get mixed together. Some Shantani is saying something. Yes, ma'am, that's true. After visiting doctor, half of the ailment vanishes. True. Just like that, your sir, Aditya sir was having some leg sprain, and then we were putting medicine and having it, and then it was relieving him slowly. And then the other day we went to a physiotherapist, and then after coming back, he's like, no, my half of my pain is gone. So it is a lot psychological. So when you are trying and testing a drug, so you cannot let the psychological bias come into the picture, both for the the investigator, who is the doctor, and both for the patient. Okay, so we have to have the real raw data, that if the drug, the medicine is given for so and so reason, then it is actually affecting it or not. So that needs to be known uh, with a bias free manner. Okay, so I cannot say that, oh, I think the pain is less. There is no place of I think the pain is less. Either the pain is less, or uh, pain is more, or if it is reducing or it is not reducing. Okay, just a layman example. So to do that, we have to you know hide the information. And as I have discussed previous day, that there is always a control group versus a treatment group. So control group receives a placebo treatment, basically not the drug that we are testing. It's a non-therapeutical effect substance that they generally, the saline water maybe you can be injected, versus, so that this is my control group, in respect to which I will compare my treatment group. Okay, so I'm giving my control the placebo, and the treatment group, I will give the drug that we are investigating currently, and then we will compare the results, then the normal versus the treatment effect, that we will compare. So for that, how do you decide, like if you remember when I don't know you guys, so it's a lot of changed situation. When we were kid, we used to play different outdoor games, and then you have to divide into teams. So how do you divide into teams? Suppose there is a football match going on, and then how do you decide that who will be in your team? So most likely, if you are allowed to choose that, okay, you are the captain, and then you choose your team. Okay, there are 10 people, it's a mini match in your uh, area or location, you are playing in the field with your friends. So if you are 10 people and you are the captain of the team, and you are asked that choose the team, so what you will do, either you will choose your friend who is great, good with you, or what will you do, you will choose the best player. Okay, so if you suppose you have the advantage of calling in first and then choosing first, then there is a will you be fair? You will always try that my team will be strong, I have my friend in my team, I have the best player in my team, so that I have a strong team. Okay, and then whatever will be left will be on the other team. So will that be fair? So when you are playing a game, and if you want it to be fair, then it has to be random. You cannot choose, pick and choose. Okay, so we used to do in when we were kid, so one person will stand, and then he and then on his back you will pat, and then you will see you will put finger one, and then who is number one, and then going to team one, and then two going to team two, like that. So this means you are choosing randomly, you are not choosing out of your preference. So similarly, when you are doing in the clinical trial, we have to do it randomly, so that just like when we are randomly choosing the players, there could be a few good players also in the team, there could be a few moderate to bad players also. So it's a mixture of both, right? So if you choose randomly, then one team will not be superior to the other. Okay, so every both the teams will have equal strengths or weakness. So the similarly in clinical trials also we are trying to do the same, that we want to divide the control group and treatment group. Maybe suppose that somebody is more ill. Okay, or somebody is a healthier, like even though suppose there is a cancer patient, but then the severity of it, or maybe a migraine patient, but the severity might differ. So if you take a less severe patient and then treat the patient and get great results, but that will not tell you that if a severely affected migraine patient, how the drug will be doing on a severely affected migraine patient, right? So you have to have in your group a severe one also, and a moderate one also, and a starting one also. So you have to have a balanced group, and so that you reduce the bias and do it fairly. So basically we want to do the trial fairly, so that we can test in all ages, all gender, all the whatever the sites we have, so different localities. So we want to have a holistic approach and get a review, that if I am here sitting and taking a paracetamol, it will affect me. But if I have a friend in Nigeria, and then that person is also taking a paracetamol, or maybe if I am of certain age, whatever is affecting on me, if a 18-year-old person is taking the medicine, if a 60-year-old person taking the same medicine, whether if that will affect them equally, with equal effectiveness, that I need to check. So for that, if I choose and pick and choose my group, whether it is between 18 to 40, then I miss on the 40 to 60 age group. So I have to choose the patients randomly, so that in my team I have all the different kind of people on which the drug that I am testing will be tested, and the blinding will be done, so that I don't think that it is affecting me. I will be telling that yes, either it is affecting me or not. There will be no psychological aspect associated to it. That is why randomization and blinding is very important. So we will go through the slide and see how what it is saying. um okay, just a minute. Koel. Hi, Koel. I see this is a new name. Shantani. I read Shantani's. So, so Bhaskar. Hi, Bhaskar. Good afternoon. It's morning for me. Good afternoon to you. Are you a new one, new student, or old one? Please remind me. I'm not sure. I've seen your name on the previous class. Even though, if even so, please remind me once. And I think Koel, you are a new person. So please introduce yourself, or where are you from, what are you pursuing, and why are you in the class, or what is, how do you know, get to know about the class. Please let us know and we will continue. Okay. So what is it is saying that randomization is clinical trial, in clinical trial is a process of assigning participants to different groups. That I said that we have to divide in different groups and it has to be random. Okay. And using a random method like a computer generated list or a random number table to eliminate selection bias, so that we don't pick and choose, or maybe sometimes, it really hampers the integrity. Suppose you know somebody who works in a hospital, and then there is a trial going on, and then you have, we have the tendency that if you know somebody and we want to take advantage that, somebody works in a hospital and they will help you get an appointment and stuff like that. So similarly, suppose somebody is working in a trial and you know that person and you want to get into that trial, and then you, now if you are into a trial, not necessarily you will get the treatment. You might get the placebo. Now suppose, I understand that, suppose somebody is in a critical condition and they want to get the treatment. So if you want to, back channel and try to get the treatment, then that will hamper the integrity. So as we all know that this clinical trial process, the clinical research, everything is a very ethical process. Okay. Because we are dealing with human being, their lives, life altering data, um, life threatening diseases. So we have to be very ethical. So whoever is wanting to go into this field and work, so you have to remember one thing, that when you are working with a data, it might be a number that you're saying, the subject number 00123, but it could be a number for you, but this is a real person back in behind that computer screen of yours, whose actual life is depending upon your judgment. So it has to be, have to be very ethical, and always have to follow the SOPs and whatever the rules and regulations that are in place. That's why it is, this industry is so regulated and regulatory. There are different regulatory bodies who monitor this, because of this reason, that you cannot be unethical and compromise a patient's safety, compromise the integrity of a clinical trial. Because suppose, as I was saying, that somebody and you are trying to get in, and then somehow through the back channels you are trying to get the treatment. I get your point that you want to do good for yourself, but if you see the bigger picture, then you cannot do that, because then you will compromise the integrity of the clinical trial. Because what we are trying to achieve here is to see in a wide population how it works, and then if I temper the data, temper the process, then the objective of running the trial will not be the same. Okay. And whatever data or inference that we are deducing is not 100% true, then the entire process of doing the clinical trial, as I mentioned and discussed, it's a very costly affair. It requires a lot of people, a lot of time commitment. So if you are not ethical, whoever in whatever roles or whatever capacity is associated with clinical trials, clinical trial process, if that person is not following the regulations, if not ethical, then you are hampering the entire process. So these things, maintaining these things, these are very simple things, but maintaining these things is very important. So we will go ahead with the slide. So what it is saying, without randomization, what would be the potential problems? Okay, so as I mentioned, maybe a researcher might feel bad giving a placebo to a very sick patient and therefore assign them to a treatment group. That here, because of this, the blinding comes into place. Okay, so if you don't know, so first of all, the idea is you have to divide randomly. Now somebody is dividing, right? Maybe I am in charge and I know that who is getting what. Okay, now me being a human being, and I know that this patient has come and really bad condition, old, so I might feel bad, and then I might want to give them the treatment. So for these reason, what, there are kit numbers, and it is just a number and you don't know what it is there. So I'm combining the blinding and the randomization because it goes hand in hand. So you have your patient needs to be divided randomly and given the treatment or assign the treatment blindly, so that you also don't know, whoever is assigning the treatment, they also don't know what is what, so that the integrity maintains. So the researcher, or maybe who is assigning, might feel bad giving a placebo to a very sick patient and therefore assign them to a treatment group. So that is not correct. So that's why we need to do it randomized. So I will tell you how the randomization happens. It's through mm, interactive process. Just a moment. Yeah, so it happens through an automatic process, so that there is no human intervention, because wherever there is a human intervention, there might be chances of human error. So this randomization is done through a different process where you can remove the human bias. So what says, or they might assign healthier younger patient to the treatment group, thinking that the drug will work better and results will look good. Okay, so everybody wants to, nobody wants their drug to fail, nobody wants to fail, right? So and this is a trial and error process. So now if you are not having the good intentions and you want to just pass your drug, so if you have the chance to choose the healthier patients and the younger patients, where the probability of the drug working fine is more. Okay, so maybe if a drug is on the third stage, and it might be the effectiveness might be less, but then if it is in the early stage, then it might affect well. Now if you have the chance to choose, just like your team members in your football team, that if you have the chance to choose the good players, then you will always do, and then your results will be better. But then in clinical trials, that will not be ethical, because when your drug is out in the market, then everybody, you cannot control that, or you cannot control that only healthier people is taking your drug. It is to cater all the populations. Okay, so if you are taking only healthier and younger people and then saying that it works 80% of its efficacy, but then that will not be true, because you have not tested on a older person or of a different community, or a different age group, or a different sex person. So for that they might assign so for that reason also, to eliminate this, you need randomization. This leads to imbalanced group, and any observed treatment effects may be due to differences in the patient, not the treatment itself. Okay, so that's what we are trying to achieve, that if we are saying that this treatment works, then we must know that we have tested in all different kind of people possible, and then this is my result. So to ensure all these things, the randomization of participants is necessary. Now let's see what happens if if a trial is randomized, then what are the benefits that we can get. So randomization generally happen through a computer algorithm or random number generator who decides that which group who goes in which group. I'll explain how it is done. The researchers had no contact over the assignment. Okay, the researcher meaning here they are saying researcher, but then it means that whoever is assigning, who is in charge of the assigning of the drug or dividing the people or enrolling the people, so that will person will have no control. So on an average, older and younger patients are equally distributed, sicker and healthier patients are equally distributed. Known and unknown confounding factors like smoking status, body weight, genetic differences are more likely to balance out. Okay, so if you have a wide range of people, then you have a variety of people with different conditions and different lifestyles and different age. So it's a good group for you to test your product. And then now we will see how this randomization happens. Okay, so if you can see, yeah, in this picture, so how you know how you get go to a bank and then get a token, right? So similarly, um, randomization also happens automatically, okay, from with an automation system that is called generally it is called IRT, that is interactive response technology. How you go to a bank and then there is a token machine, and then tell you it's an interactive session, right? So they will tell you that now enter your number. Or so for a bank token it is fairly simple. Or when here we go to the hospitals, so you can self check in. So what happens, I'll give you a scenario. What happens when here in UK we go to the hospital, and you have to self check in. You have to tell them that yes, I had an appointment, but I have reached, so that they will add you to their queue, and then they will call you. So there is a tab where you can self check in. So it's an interactive process where it will ask you your name, your date of birth, and I think few other details also they ask. And then they will check in that what time you have reached, and then it will tell you that how long you might have to wait. Okay, so if you have go to an OPD, then it will give you a approximate time that there are 13 people ahead of you and you have a half an hour wait time. So it gives you a information, it's a clear picture. Okay, so but the clear picture is not our concern right now. Our concern is this system that is allowing you to give this, okay, this token number, and then it will give you a number that you are on the 14th in the list, and then they will display your name and stuff like that. So like that it is randomly done. Or just like when you go to a bank and purchase your not purchase, you take your token number, and once your token number is coming, then you go. So there is no preference, okay, that the bank manager and then you will get ahead in the line. So whoever has come in before you will go before you, and then accordingly it will happen. Similarly, in this IRT system, it is integrated to the EDC platform when the study setup is happening. Okay, we will learn setup and how all this integration does happen. But then for now, for our overview, so this in that interactive system that gets integrated with our trial basic knowledge. And once a new subject comes, and then they will take your vital signs, and if you are eligible for the study, because as we discussed previously, that there will be inclusion exclusion criteria based on which you will be judged whether you can be in the study or not. So if you have crossed the inclusion, like you are eligible for the study, then you get randomized. Okay, so what happens, there could be a computer system also, that you put your data, that your name, your date of birth, your sex, and all these basic informations you will give, and then based upon which you will get a number, randomization number. So the system is integrated in such a way that it will divide you. Suppose we are denoting the control group as group A and treatment group as group B. So it will randomly uh, assign you a group. Okay, so based upon, you just have to put your details. So this can be a telephone, like telecalling operations also, wherein you dial in, and then, or the site people who is doing the randomization, they will call and then provide the details of the subject, and then based upon the subject they will give a number, like suppose the study number is P31, and then the subject number is 00032. So it's a whenever we work in the database, so subject numbers comes like that. So in that, there is different formats through which, like, what we need to follow. So in your randomization number, that subject number that you get there, you get the subject, the study number, the site number. So many a times the studies are multi-site, like there are different site, different in different hospitals the trial is going on. Okay, so if there are 5 sites, then there will be different numbers, like site 1, 2, 3, 4. So when the randomization is happening, then the subject, in the subject number, the site code also, it depends how you have created the or how you have integrated the EDC system with your IRT, and then it will give you a number. Okay, and similarly, to keep it for going on with the blinding, that you don't know which if you're getting the treatment or the control group, similarly through this IRT system you can get a kit number also. So you only know that, so when the site person will give you the treatment, so they will only see the number, kit number, and the sponsors also send the kit with the kit numbers, and then they will just give you. So the who is pushing, suppose you are supposed to take a medicine through injection, so the person who is giving you the injection, that person doesn't know whether it is a placebo or a treatment. Okay, and you also not getting to know that whether it is a treatment or placebo. You just have a kit number, and looking at the kit number only, the site person also, or the nurse who is pushing you the injection, so they will give you. So nobody will have the knowledge, it's all blinded, that nobody will have the knowledge that what is going on inside your body, so that you do not have a bias that, oh, I got this medicine, then I will start feeling better immediately, or I received the placebo, so no, I'm not feeling well. So okay, so to ignore all this bias, through this IRT system we get the randomization number also and kit number also. So this is overall the randomization process, and then we'll similar along with this randomization we will see the blinding also. So what happens, there are study design depending upon the requirement of the study, it could be double blind, single blind, or triple blind. Okay, so if it is a single blind study, it depends, and it is decided by the sponsor, that what kind of a study they want to run. So if it is a single blind study, then only the patient will be blinded meaning they will not know what they're receiving. If it is a double blind, then the doctor and the patient both will not know, because many a times if the doctor knows that what it is get what get, what the patient is getting, then he might also expect a certain kind of response from the patient. So all this psychological aspect, you might say that, ma'am, that is not logical, but you know what, human being is human being, and it's not always logical. You don't operate, humans don't operate with logic 100%. So there is other factors that come into place, and it is bound to happen that the psychological aspects come in your way. So to eliminate all those things, mostly studies you will see will be double blinded. Okay, so in double blinded situation, the doctor will also know what the patient is getting, and the patient is definitely not knowing that what they're getting. So blinding in clinical trials is a method used to prevent bias. Again, what we are discussing is on the same line, that randomization and blinding both happens to eliminate or prevent the bias by keeping certain information hidden from one or more patient parties involved in the study, typically patients, doctors, or data analysts. So in terms of if it is the third blind, so that is a little rare. Generally studies are double blind, but then many a times, uh, if you want to further protect your data from any manipulation, then you might want to blind your data analyst also. The goal is to ensure that the treatment's effect is evaluated objectively without influence from expectations or personal opinion. Okay. Okay, Basudev is saying something. When both doctors and patient don't know what the drug is, what that is given, it is called double blind. Correct. So the main purpose is to evaluate the effect of the drug objectively, and not from any expectation that I have taken this drug, then it should lower my uh, symptoms. It should, but whether it is or not, that we are trying to know this. So that's why we cannot have an expectation. We have to be bias free. Okay, why data blinding is done? To avoid placebo effect. If a patient knows that they are receiving the real drug, they may psychologically feel better. Okay, reduces observer bias. If a doctor knows which treatment a patient is receiving, they might interpret symptoms or outcomes more favorably or unfavorably. Okay, so doctor might think that, okay, I have given the medicine, that's why it is behaving a certain way. So that could be misinterpreted. Ensure data integrity. Data analysis could be consciously or unconsciously manipulate results if they know which group is which. So as I mentioned, that if you further want to mask your data from any manipulation, then mm, the data analyst might also get blinded. Okay, so this ensured that the reported side effects, immunity levels, or symptoms were unbiased. Okay, so suppose you are taking a medicine, and if you know that this will reduce your pain, then you might also, sometimes it might not always work in your favor. It might give you trouble also. So, because if you know that you have taken the medicine, then you might ignore some signs also. Suppose you have taken the medicine and it's still not affecting you, but then psychologically you will think that okay, I have taken the medicine, it will help me, let me wait for a while. And then maybe the judgment will be hampered, that when suppose you are supposed to tell that after taking the medicine within 1 hour whether if it is affecting you or not, and what is the you know grade of its effectiveness, you might get judgmental that whether, suppose you are asked after 1 hour of taking a medicine, if your pain was 10 on 10 initially, then after taking the drug, what is the degree of intensity. But then if you do not know that what you have taken, then you will tell the real number, that if it is still 10 on 10 or it is little less, it is still 9 on 10. But then if you know that yes, you have taken the drug, then you might psychologically get judgmental and think that no, I think it is fine. So overall it will get hindered. So it is better that you do not know what you have taken, so that the result comes clean. Blinding ensures that the doctor don't expect better results from the new drug and record subjective symptoms more favorably, the same thing that we are discussing. So the types of blinding are single blind, only the patient is unaware of the treatment; double blind, both the patient and the healthcare provider researcher are unaware; and triple blind, patient, researcher, and the data analyst or statistician are unaware. So depending upon the requirement and the sponsor's decision, what they're how they're wanting the data and how much data protection they are trying to achieve, depending upon that, the study design, they provide the study design. Okay, so this is our randomization and blinding concept that is very 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 important in our clinical trial process. Okay, so if you have any question regarding randomization and the blinding process, you can ask me here, and we will discuss that. And next we will briefly discuss what are the employers, what are the companies are there who is giving us the job, who can be a potential employer for us. Okay, so if you have any question, 9 of you who are in the class, shoot. Okay, Jagdish is asking a question. Jagdish is asking, is randomization done in all phases of clinical trial? Good question, Jagdish. I should have mentioned it. Randomization is the very first thing that you will do once you are getting into the trial. So, how it happens? Um, a trial is in the go live phase. Okay, now we will be enrolling for the trial. Now a patient is coming, it came to the site, and I'm telling you the process flow. So I'm a hospital, I'm a site running so and so. Suppose I'm running a diabetes trial. Now a patient has come, and then I will check their vitals, and then I'll see that if the inclusion criteria is matching and the exclusion criteria is also matching, that whatever criteria I require for my patient to have to get enrolled into the study, like if it is a diabetes study, then the patient needs to have diabetes. And then if I have some age limit, like I only have need adults, then the patient definitely has to be more than 18 years, and so on and so forth. There could be many other inclusion criteria and exclusion criteria. So everything if matches, and the that person is eligible to get into the study, and then they after the eligibility criteria, they sign the informed consent, as we I hope you remember that we discussed about the informed consent form. So after signing of informed consent form, then randomization happens. So once the randomization happens, then they, the patient will enter into the study. Okay. So randomization and the date of randomization, when you will actually work in an EDC, so you will get a form where randomization details are there, and so these basic information will be there in the form, whether eligibility criteria, it will be written yes eligible or yes uh, randomization, and randomization date, and the date of randomization and informed consent date, that could be same, that today I'm giving informed consent for enrolling into the study, and today only I'm getting randomized. That can be done. But then these are the two process after which randomization is done. And to give you an overview of the study overall, so when a study is happening, okay, so first will be in clinical data management. As a clinical data manager, um, okay, so as a clinical data manager, randomization will happen when the uh, study is in a conduct phase. Okay, so once the protocol is taken from the sponsor, and then everything is done, and the EDC is created, database is created, then when the patient enrollment is happening, at that point the randomization will happen. Then once it is done, and then with those numbers, those randomization number and subject number, the subject will continue throughout the course of the study. So it happens in the very beginning of the study. Yes, good summarization, Yasmin. Randomization and blinding is necessary to remove psychological aspect. Yeah, any psychological aspect, any preference. So we cannot prefer people. Okay, so we have to be fair and just with the and follow the regulations of the trial. So for that we need to do randomization and blinding. Priyanka is asking, ma'am, please repeat ensure data integrity. Uh, Sorry, Priyanka, I did not get your question. Could you elaborate your question? What you were wanting to know? So, if I'm understanding it correct, you want to know that how, if we are not doing the blinding and randomization, how data integrity might compromise, and how we are protecting the data integrity. So suppose if you are not if you are excluding a person, a section of people in your trial. Suppose you are only taking 18 to 40 age group, and maybe you are only taking male subjects, or maybe a specific area, then the effect of your drug will be showing in only that category of people, and then you might get a good result. But then suppose you have constricted and you have a small number of like not diverse group of patient where you are testing your data, but then once you are getting the output and releasing the drug into the market, but then there you are not controlling, you got your good results, but then now everybody is taking the medicine. Okay, but then how your integrity is getting compromised, right? Because you did not test your drug in a wide range of people. You have chosen a best group, and then did your trial, but then now you're claiming that everybody can take the drug. So there is an integrity issue, right? So you have to be fair and just and honest and truthful about what you're doing and what you're telling people. So is that answer your question, Priyanka? If not, then please tell me what you are exactly wanting to know. I'll explain you. Rishi is saying something, ma'am, in phase 2 and phase 3, the number of volunteers also increased. So randomization is done each time there is an enrollment of new volunteer in each phases of clinical trial. Yes, yes. So each phase is a wrong term. So yeah, so whatever may be the phase, if it is phase 2, then you might have the so and so number, it could be in hundreds, and for phase 3 it could be in thousands, but randomization is 100% necessary. So that is the gatekeeper. So through randomization you get inside a study. Without randomization you cannot just start your study. You have to have a you have to go through the randomization, you have to have a patient number, your kit number, and then you are onboarded in the randomization. Is the process by which you get onboarded in the study, no matter what is the phase. Okay, is that clear, Rishi? If not, you can ask more question. One more thing, in phase 4, that is in post market surveillance, we do not perform randomization, right? That's correct, because it is out in the market. So that's a very good question and gives us a point to discuss that even though we are doing randomization and trying to make our trial as diverse as possible, still, when you are doing the trial, if it is phase 2, phase 3, you can check 100 to 1,000, maybe it could be 2,000, 3,000. I have worked in a study I used to handle. It was a maintenance study for cancer patient, for multiple cancer. It was a 20,000 patients, but still 20,000 versus when the drug is out, it is millions and billions of people, everybody out there. And then there could be one person who can have some problem with it. So you cannot 100%. So to answer your question, Priyanka, more elaborately, that even though we are trying to make our trials diverse, still there is a chance that we miss something. So that's why the post market surveillance is done, where we keep a track. So there we cannot because when I'm doing a trial, it's a controlled format, right? So I know that these patients are coming, I can control the population. But when it is out in the market, then I cannot control who and who is taking the medicine. So I'm keeping a track of those people, but then I cannot do the randomization. So it is automatically getting randomized, because anyone can here I am giving permission that okay, you can be in the trial, but when it is out in the market, so it is automatically random. You can also mm, Rishi, you can also have the medicine, I can have the medicine. You are from a different age group, I'm from a different age group, you are male, I'm female. So it is automatically randomized, and that's why we keep that data. And then depending upon the sponsor and how long they want to check and keep the update, so that is the reason post marketing surveillance is done, to get a bigger picture of how the medicine is doing in the market. Okay, I hope I have answered your question, Rishi. If you still have questions, you can ask. And Priyanka is saying understood, ma'am, that's great. Okay, so if you have any more questions, please keep on asking. If not, then we will take 2 minutes, 5 minutes break, and then we'll go to our next session. If you guys, not everybody knows that we have rescheduled our yesterday's class, so we are taking 2 class back to back. So okay, great, Rishi. Rishi is saying yes, ma'am, understood. That's great. And you can still keep thinking. You take 5, and then we'll meet in 5 minutes, and then we'll continue our class. In between, if you have any more questions, please ask. We will discuss. Okay.
IASD করছে জীবন কেটে বেকারত্বের জ্বালা, চাকরি জীবনের কাটতে রিবন, চলে এসো এই বেলা, চলে এসো এই বেলা। দিনকাল একদম ভালো না, চাকরি আজ খুবই রেয়ার, প্লেসমেন্টের দায়ভার আমরা তাই করছি শেয়ার। IASDর হাত ধরে হবে তোমার প্রথম রিক্রুট, তাই সব টেনশন যাও ভুলে, জীবন এবার হবে রিবুট। IASD করছে জীবন কেটে বেকারত্বের জ্বালা, চাকরি জীবনের কাটতে রিবন, চলে এসো এই বেলা, চলে এসো এই বেলা। ইংরেজি টেকনোলজি ভয় পাও বুঝি, হবে এবার সোজা, যাবে কোথা খুঁজছো বুঝি, এসো এখানে শেষ হবে তোমার খোঁজা। IASD করছে জীবন কেটে বেকারত্বের জ্বালা, চাকরি জীবনের কাটতে রিবন, চলে এসো এই বেলা, চলে এসো এই বেলা। জব গ্যারান্টি সার্টিফিকেট সবই চাও বুঝি, ভুলে টেনশনের বোঝা। IASD ঠিকানা তবে এখানেই শেষ পথ খোঁজা। IASD
अनगिनत एहसान है मेरी सांसों पर तुम्हारा, अनगिनत एहसान है तेरी खुशहाली रहे, कहता मेरा ईमान है। मेरी सांसों में तिरंगे की बुलंदी की दुआ, मेरी सांसों में तिरंगे की बुलंदी की दुआ। मेरे हिंदुस्तान तुझ पर जान भी कुर्बान है, मेरे हिंदुस्तान तुझ पर जान भी कुर्बान है।
প্রতিশ্রুতি নিরাপত্তা জানি আজ খুবই বিরল, কথা দিলাম আমি তবু আজ পথ হবে খুবই সরল। প্রতিশ্রুতি নিরাপত্তা জানি আজ খুবই বিরল, কথা দিলাম আমি তবু আজ পথ হবে খুবই সরল। সহায়তা পাবে সবটা, প্রদর্শক অতি অটুট।
Hello, welcome back. I hope everybody is back. Join back quickly, we will start. Okay, so whoever is out, join back quickly. We will start our discussions. So far I want to tell you that so far whatever we have discussed, these are your basic material. We will whenever you are going, hi ma'am, hi Priyanka, hi, welcome back. So these are the topics that you need to prepare for your interviews. Okay, so during our course we will get to know about the processes, how it is done in detail, but those are the things to better prepare you once you are getting and joining a company. Okay, because they will anyway train you, but then I am training you ahead so that it will uh, position you in a better way, that once you are going inside and get the job, then you will know about these things, you will have a basic information how all the activities is done. Okay, so that is a plus. But then until this point, or until not this point, this class, the currently the class we will be having, and next we will learn about the regulations and uh, I say GCP guidelines and all those stuff. So these are your preparation points as a fresher when you are going for an interview. Okay, so all these control group, placebo, what is protocol, what is interim analysis, what is randomization, blinding, and then the phases of trial, how many patients are involved in this is a very common question. Like this is this is will be definitely be asked that what are the phases and how many people are involved in which phases, and what do you achieve in each phases. Okay, what do you do in each phases, that we have studied previously. So these are your preparation points. So please uh, be careful in these classes and make a note of these topics and prepare all these topics, because they will not expect you to know how discrepancy management is done, or how a SA reconciliation is done, or how a CRF is built. So it will come as and when you will work. It is good to know. We will prepare you for better, and then prepare you for better growth. But then these initial topics that we are talking, they you will have to prepare them for your interviews. Okay, so be mindful of these classes, make a note of each topic, and then prepare for them, because they will get you into the job. Okay, so now before we get into the roles of clinical data manager, I wanted to briefly tell you uh, just a, I hope everyone can see my slides, right? I think the slide design is a little different. If you have any issue, just let me know. If you see fine, then that's all right. So I wanted to discuss you example of the employers that we can have. So there are three broad categories where we can have jobs. Okay, one is, everybody knows, clinical research organization, CROs. Second is the BPOs. The generally we know about BPOs like TCS, Accenture, these companies. And three is product based companies. Like there are companies who are creating medicine, so who they are the medicine company, and they also have their own data management unit. Okay, so they do not, some they also work with CROs, but they also have their own facility where they do their own clinical trials also. So if you can see in this, there is a lot of names. Okay, thank you Basudev for confirming. So in there is a lot of names. I will divide the names. Like in our CRO category, you know, it will come as IQVIA, Icon, Seniors Health, and then you will have PPD, you have, I think, what are the other names are there? So yeah, so these are the names that here PPD, Paraxel, Okay, PPD, Paraxel, Icon, IQVIA, these are the companies. They are your CROs. Okay, now second category is BPO. So in BPO category, we have TCS. TCS have a life science wing. Accenture, Cognizant, they all have their life science wing, and they also have life science client. So they also do clinical research, like clinical trials. So CDM roles are available within them also. And there are product based companies like Novartis, Novo Nordics, now GSK. So all these companies, Roche. Okay, so all these companies, they also Johnson and Johnson, they also work with CROs, but they also have Pfizer also. Pfizer, J&J, all these companies, they have their own setup also, where they also do their clinical research. So you can these are your reference. So you can look for your opportunities in naukri.com, and then LinkedIn, and then other job portals, and you can individually go to their sites also. And then there is, in every company, you will have, if you go to the website, you will see there is a careers platform. So if you go into that, then you can see their latest whatever their vacancies are, and then accordingly you can apply. Yeah, these are the few names. I'm missing a few more. Paraxel is there, PPD, Icon, IQVIA, Seniors Health. I will give you a detailed list, okay, uh, for the CROs. I'll share the information with you. But then in general, you can see in Naukri, and I have discussed with you what are the roles available. And for clinical data management, as I mentioned before, role names can vary. It could be clinical data coordinator, or clinical data associate, or clinical data manager. Okay, these three roles names generally appear for our CDM roles. Okay, so if you are looking for a CDM role, then you try, because different company uses different names. So you might be trying for a clinical data coordinator, and somebody who is referring it as a clinical data associate, you might miss that. Okay, so all three names you can through all you can filter your search in from these three names. Okay, so we will go to the next slide. So what is the role of a clinical data manager? Okay, what it is done. So broadly, clinical data management people, they do data collection and capture, data validation and cleaning, data integration and management. We will go one by one and discuss what all these are. Then data design and maintenance, data analysis preparation, regulatory compliance and reporting, and project coordination and communication. So these are few high level headers, and then we will go one by one and see what it means. So data collection and capture, designing CRF forms, paper or electronic, to collect patient data systematically, ensuring data is captured according to the clinical trial protocol. Okay, let me give you an overview. Okay, so what is the role of clinical data manager? So basically, clinical data manager is the gatekeeper of all the data that we are collecting from the site. Okay, so all the data that we are producing in a clinical trial is technically with clinical data managers. Okay, through the EDC system, electronic data capture system. So simple. So we are running a trial. we have the patients, we will collect the data. So the data needs to be captured somewhere, right? We have to write the data somewhere. So previously it used to be paper trials where there were paper CRF, report case report forms, where all the details of the patient would be written. Now everything is electronic, so we have ECRF, which is built in EDC system, electronic data capture system. Okay, so if you see from a clinical study point of view, what we often say that we do end-to-end data management. Okay, what does this end-to-end data management means? So though let me mention you that when you enter into the workforce as a clinical data manager, CDC or CDA, you work in the s- okay, I think I'll give you a better overview. So, okay, for a study, clinical study, it is divided in three phases. Okay, setup, conduct, and closeout. Okay, in setup, what is done, that these ECRS, CRS, and in EDC system is getting built. In the closeout phase, all the data gets captured. So whatever data that we need to capture, all those forms we need to create. So in setup phase, all those forms get created. Now once the forms are created and ready, then we will do the data capture. So when you are a fresher and get into the industry, you will generally work in a conduct phase where the data is getting captured and you will do the validation and data cleaning. So these are the process initially, because the data the designing part is a complicated part. It needs experience and knowledge. So as and when you get the opportunity and you get experience, then you move to the design part. But initially, when you are a clinical data manager starting your career, you will generally work in conduct phase and then close out phase. Okay, so what we do in this conduct phase and close out phase is data validation and cleaning. Excuse me. So as we know that in clinical trials, we work as per the protocol. So whatever data that is getting collected from the site, we need to validate those data, that if the data whatever is getting captured is as per the protocol or not. If there is um, any fault in the data, if there is any defect in the data, so that we need to be checking. So that process of checking the data whether it is correct or not. If it is not correct, then asking the site people, the CRC, that this data seems like a wrong data. You check in your database or you check in your source data. Did we talk about source data? I think we did, but then I will again, just for your reference, that source data is when the patient is in the hospital and suppose a blood is taken for some analysis. Okay, so there will be a form, and then if you I'm sure you have at some point in your life you have went to a hospital and you see for the nurses, they always have some papers and they're always filling up something. So what they're filling up, all the details that they have done. So as soon as you go to a hospital and maybe you have something, so they will take your BP, they will take your temperature, they will height, weight. So this is called vital signs. And then suppose they are drawing some blood, then they will have a form. So what sample they have taken, how much a quantity they have taken, for what analysis they have taken, what is the time, what is the date. So these are on that point, like on the spot. um these information is getting recorded. So this becomes your source data. Source means this is the original data. you are directly interacting with the subject, and whatever information you are getting from the subject at this moment, real time, you are capturing this data. So this is your source data. Now this information gets transferred and updated through data integration to the EDC data, which comes to a clinical data manager to review. Now suppose this data is coming to you and you see there is a problem in the data. It could be anything. We will discuss what kind of problems that might come when you review a data. It could be a missing data. Sometimes when the person who is doing data entry, they miss to put the date. Okay. Or they miss to update the date, they miss the age. Or maybe sometimes when it is I'm sure you have all seen blood reports where you get ranges, like if it is a RBC count, then you have your result that this much you have, and then you get a range that it should be the normal range from 0 to 1 or whatever the range. So you know the data discrepancy could be many forms, like maybe they have missed update, they did not put a, how do you say, so they missed an parameter to draw the blood, or even they have drawn the blood but then they did not mention in the form that they have drawn the blood. So these kind of very different kind of anomalies you have when you are checking your data. So if you see any uh, fault in your data, then you can query it back. There in our EDC system gives you the option to write it to them, the site people, that maybe this date is missing, so please check in your source data whether the date is present or not, or the age is missing, so please check that whether you have. So sometimes it is just a data entry error, that it is there in the information is there in the source data but they missed to update it. So in those cases they just update the data. Sometimes what happens when suppose there is an influx of a lot of subjects and then they missed taking the data, they missed updating the date. In that cases they will come with an answer that date is unavailable. Okay. So in that case, we have there are parameters where if the date is unknown, then you can, there are formats you can use, like UNN. So these kind of information are allowed, that if it is unknown then you can mention it that it is unknown. So these kind of data validations and cleaning needs to be done from a clinical data manager's point of view, so that you ensure that whatever data is getting collected is correct and validated. Okay. So detecting and resolving data, so these are very simple formats that I have given you examples. There are complicated data validations and cleaning, there are in process. So for that we have checks in place, like if when your database is getting designed and created, then only there are triggers that you put, so that if some data is missing, then it will give you a query. So okay, in next class, I will try and collect some ECRF, what do you say, screenshots where I can show you that how a query looks like. So generally, when you are a fresher and you get into the job, generally discrepancy management, query management is the first activity that you are given, where the discrepancies are there, and then you have to see why the query has fired, and then depending upon your judgment you have to close the query, or if there is an update then you have to check that if the update is fine or not. So these kind of things. Okay. So basically there are complicated data validations and cleaning processes through which you will be cleaning the data, and in different frequencies, like if it is a discrepancy management, query management is done on a regular basis, and then there are other kind of data cleaning process which is done on monthly basis. Okay, so overall the data validation and cleaning is the very important aspect of a clinical data manager's role. Okay, so detecting and resolving data discrepancies or errors through queries, validating data to ensure completeness, accuracy and consistency. So there are many times we have missing data, we have missing pages, maybe a visit has done, but and as per protocol and schedule we are expecting the data, but then it is still missing. So we have to query that the visit has done on so and so date, but the date, like, the visit date is past, but then data is still not present. So you, in that cases, you have to check the data completeness. Okay, so suppose today is 18th and a visit was scheduled on 15, but then there is a 3 days leverage time, then within 3 days the data needs to be updated, and then by 18th you still see that the visit date and rest of the details are not present. Then you can query to the site that my this page is incomplete but this is expected. So you are also checking for the completeness of the data, accuracy of the data and consistency of the data. Okay, and then performing data cleaning activity to maintain high quality data. So that is always there through different activities. We always check and clean the data so that the data quality is maintained. Okay, because this is the data that as clean as we have the data, that will determine the integrity of the study and the effectiveness of the study. If wrong data is updated, then the overall trial can go for a toss. So we have to be very vigilant about the data that is getting entered. It needs to be correct. Okay, so this is initial work will be your this, and then you will eventually move to the design part where the crfs are designed, ecrf are designed, so that the data can be, so the data is getting collected, but we have to ensure and design the form in such a way that whatever our protocol is required, like as per protocol, whatever data that needs to be captured and analyzed, so we have to design our crfs in such a form that all the data can be captured properly. So that is also an activity that a data manager does, but down the line, maybe 3 years or 5 years down the line, but depending upon the opportunity that you are getting. But then you should always look out for, once you are in a company, you should always look out for, and once you are the role that you are doing and whatever activities that you are given, you are mastering, you simultaneously, whoever is your lead, whoever is above of you, you must ask to ask them to teach how a data design. If your senior is doing a setup activity, then you might ask them that groom me or teach me. So these kind of things you can tell, so that, because that designing is a very important part of the data management's role. So it will be good that if you tell your seniors that you want to learn, and then whenever you get a chance, you learn how the designing part is also done. But that is also a very important activity that a clinical data management manager does. Okay, next is data integration and management. Okay, so integrating data from multiple sources like labs, images, patient diaries, managing large volumes of clinical trial data efficiently, ensuring compliance with CDISC. Okay, CDISC we will skip for now. That is that is kind of a programmer's point of view, but we will understand these first two points now. Integrating data from multiple sources. So what happens, as I mentioned, that when a trial is happening, so the patient is tested holistically. Okay, so and the site where the study is getting performed, so the sometimes the infrastructure of that hospital may not be enough. Okay, so maybe there are 10 lab tests that you need to do, but then in your facility, in your site, only 3 of that, those type, like 10 analysis you need to do, only 3 can be done in the hospital, because the other 7 you don't have the infrastructure, you don't have the machines. Not all the hospital have all the equipments, right? So sometimes you have to go to another diagnostic center to do some tests, right? So these kind of data needs to be integrated into the EDC system. Like you can go for a scan, you can go for a lab test, you can patient diaries, so many. What are data cleaning activities? Rishi is asking what is data cleaning activity. Okay, Rishi, I'll tell you in detail. We will get through this point and then we'll go back to data cleaning once more. So so as I was saying, that maybe your site has equipment for 3 tests, but then rest of the 7 tests you need to go to another lab and do the test. Okay, but then we need all those informations, whatever the test results, we need them to analyze our drugs effect. So all these data, there are processes through which the lab data get integrated. The imaging data, if I can give an example, maybe you need an x-ray, you need a PET scan, you need an MRI, okay, for whatever you have going, whatever you are going through. So those imaging data we also need. So sometimes we do get to integrate in our system. By saying integrate, what do I mean? That when i'm op- I'm opening my CRF, I'm getting those information integrated in the system and I can see in the form. But then sometimes the integration is not possible. For that, we have lab vendors through which we do these tests, and then those lab vendors give us some data. Okay, they provide their whatever their results are coming. Okay, so these kind of data also are clinical data managers purview to check. Okay, they have to for that there are activities that like reconciliation are in place, where we have to check these vendor data, that whether they are correct and they're effective or not. Like patient diaries also. Patient diary is a format where suppose you have a lifestyle disease, or maybe you have migraine, or these kind of or dementia, or schizophrenia, or any this kind of neurological disease. In that case, there are a lot of behavioral problems, or how you feel, how the pain intensity, those kind of things needs to be monitored. For that you have questionnaires. Okay, those are called as patient diaries. So maybe weekly twice you have to fill in that, if you're in a week, how many days you have pain. Maybe suppose I'm giving an example of migraine pain. Okay, people with migraine, they have severe headaches. Now not every day is same. Some day they have pain, but then they can do other works with the medicine. With their medicine, painkillers, they can do their works. Okay, but there are some days they're like inactive, they just lie down, they vomit, it's like the intensity. So what do you have in this patient diaries? The questionnaire, it says the intensity, like in a week how many days you have had pain, maybe 3 days. There could be question like, if the pain intensity, was the pain intensity similar for all the days, how long the pain was lingering. Or maybe for neurological diseases, like how are you feeling, the feeling matters a lot, how you are feeling, if you are feeling dizzy. Maybe for some studies, for some diseases people have suicidal tendency. So those are also. So all these things, patient do get some questionnaires to fill in, and then when they come for a visit, they bring in all those data. So these kind of data also needs to be assessed. So these needs to be also getting included in the ADC for a CDM to um, analyze. So these kind of data integration and management, these kind of data management also is done by clinical data manager. Now, database design and maintenance, as I mentioned, that you will be creating the database, and the design you will write the specifications, and then you will work with the other there are other people who work in data management, like portfolio in the clinical trial. There will be programmers, there will be statisticians, I have mentioned previously. So you write the specification, give them to the programmer, they program, you test. So all these designing and creation part is also part of clinical data manager's work. Okay, if I come back to Rishi's question, Rishi is asking what are data cleaning activity. So as I mentioned, Rishi, that once you collect the data, there could be a lot of defects in the data. Okay, there could be missing data also. Maybe data is present with the site people, they have recorded the data, maybe they have recorded the date, but they have missed when data they are typing the data to the EDC system, they have missed updating the data. So in that case, it will show that it is a missing date. Okay, suppose visit 1, first day of visit, or as I discussed, randomization that date needs to be there. But then suppose when you are seeing the randomization form, there you see the date is missing, and then it comes with a it's I think it's my fault. I should have uh, shown you a page. I will definitely bring in examples next day, where I will show you the screenshots, because it's a it's a little difficult to get all this, but I'll find it, and then I'll show you how it looks when a query is formed. Okay, so it comes with a red, okay, give me a moment, I'll just quickly check. uh, okay, wait. Okay, see here, just a moment, I'll just reduce myself. Okay, so this is a page of a metadata rave ECRF. This is an ECRF. Okay, CRF. It's ECRF means it's a form where you will, it's like any form when you fill your details, and suppose you are applying for a college. Okay, so you fill a form, you fill your details. It's similarly like that. It's just a form in different formats to capture different kind of data. So this is a demographic data. So for each and every subject, when they are getting enrolled, we have a, what happened? Just a moment, something happened. Yeah, so we always have a form called demographics where the date of birth, ethnicity, the gender, okay, so childbirth potential. So depends. So if it is a female subject, then childbearing potential. It is necessary for many. It's a very important aspect for a female to be updated here, because many we when we are trialing a drug, we want to know whether it is safe for pregnant women or not, okay, but or sometimes for many inclusion exclusion criteria, if it is not safe, then we cannot include pregnant women. So for that, all this information is important. So now I think I need to reduce myself a little further. Yeah. So can you see, this is how a CRF looks, okay? So ECRF looks. Now all these data needs to be here. Now can you see here ethnicity, there should have been a query here also, here also. So this is an example that suppose this form is there, but and the site has written the date of birth here, the race here, gender, and childbearing potential, yes or no, okay, everything is updated. Only they missed to update the ethnicity of the subject. Now when there are types of discrepancies and the types of checks that these kind of checks are integrated. These are called custom function checks. So these kind of checks are integrated when you are building this form, okay, that if somebody missed to update this data, a query will form. So the fundamentals of clinical data manager and the basic activity of a clinical data manager is to do query resolution. So this is a query. If a query fires, it looks like this. It's become red, and then it will it will give you a pop-up. It is saying that required field checks message. So ethnicity is a required field, okay, so it needs to be there. So now it is missing, so it is indicating that there is something missing and a query is fired. Now once you are in the company, because I cannot get a real CRF, but I will try and find the formats where I can show you that what is there in the drop down. So suppose ethnicity, there will be 5 options like Hispanic, and then there are others I'm forgetting right now. So what are the options that is available? If it is an actual ECRF, then you will be able to click in this drop down and choose one, okay. So sometimes because you have this drop down, you can choose one, and there is other also, there is always an option called others. So for other, this text box is there. So if it is other, then you can mention that what is this other. So this is how a check looks like, this is how your query looks like. So so cleaning activity meaning, Rishi, that these kind, there will be many discrepancies that is arising when the data is getting updated. So when a discrepancy or there is a wrong data or a missing data that is there, then these kind of red thing and query fires, this phenomena is called firing a query. So you have to go and check and analyze. So in this case, here is the here can you see this white field? Here you can write that you can write a query. to the site. So this is a to and fro process. If you write it here, this message will go to the site people. Okay, so if you write here that query 1, and then we have to put prefixes, and then if you say that query 1, that ethnicity is a required field but it is missing here, you will write like this: so kindly update the ethnicity, and then you will put full stop, and once it is a regular one, you will save it. Okay, so your query will get posted and it will get this get to the site people. So whoever is in the site in CRC who will resolve this data, they will get to see now a red thing like this on their end, and they will see that you are asking them to update the ethnicity. Now if they have the ethnicity, maybe they have it on their source document but they have missed updating it, then they will come and select from the drop down and update the data. Once they update the data, it will again come to you. Here the category of the query will be there. So when these kind of query where you are asking them something, it will be written here as open query. Okay, so this is an open query, and now once you are also requeried, and now if they have updated the ethnicity, it will again come to you, but the status of this query will be answered. Okay, it will be written as answered because they have put an answer. That suppose it is Hispanic they have selected. Okay, so now you have an update, they have answered your query. Now you will just cross check that okay, ethnicity was required and they have put an ethnicity here, updated an ethnicity. And then if you are satisfied with the data, then there will be a drop down here. Right now I'm not able to show you. I'll find all those examples and I'll bring it to you for your better understanding. So then you will have a drop down to close the query. So you will close the query and this red thing will go. So this is how you do the data cleaning. So to give you to explain you that how you clean the data, right? Like ethnicity was required, you saw that ethnicity was not updated, you asked the site people that ethnicity is required information, it is missing, please update the data, and then they updated, and then you close the query and clean the data, right? So it was a missing data, so we cannot keep a missing data because it is important to have the ethnicity. So this is how you clean your database. So like this there could be many different kind of checks. So whenever a query fires, it looks like red, and there are indicators from where you can know, or there are reports from you can pull from where you can see which page, because there will be plenty of pages and plenty of subjects, so you can manually go one by one and see what is the query that is firing. So there will be reports through which you can see that, see, this is the, can you see, this is the format, right? So this is the center, some medical center it is written. So this is the site name, so there could be multiple sites, so all the site name will be there. And then this is your subject number. As I mentioned, during the randomization we get the numbers. So this is depending upon the format that the study is following, we will get this subject number. So can you see there is a human type of icon that is there. So this mentions that this is a particular subject, this is a site. So can you see here is three, okay, I'll just zoom in even more further. The picture quality is not that great. Can you see here, it is showing, the icon is showing 3 human. So that means in a site there could be multiple subjects, and then you are working on this specific subject, so there is only one human being sign, and then this is a form sign, this is a demographic form, there could be multiple forms, there will be other forms also, this is one of the forms. And all these are very different things. We will discuss it as and when it is unfolding, because it will be very confusing for you if I say everything today. But to give you an understanding that data cleaning activity primarily means actioning your queries, and then there will be other logics through which you will be, those are called manual checks, or your programmed checks. So through which there will be logics through which you will be comparing data and find out if there is any discrepancy or not. So there will be few checks which will be inbuilt, and it will show you upfrontly that there is a missing data or there is a out of range data. So you can just cross check and update or clean the data. But there will be some logics with which you have to go, pull the reports and investigate whether, put formulas and see whether the data is correct or not. There could be different categories of data discrepancy. So this is how, if the data is not correct and not up to how the protocol wants it to be captured, if there is any discrepancy, then you will go raise a query, talk to a CRA, email a few people, and then clean the data so that we have the accurate information. There is no wrong information that we are carrying around, because depending upon this data will decide whether our drug will be um, passed or not, or coming to the market, depending upon this data. Okay, so it's a very important job that you are doing. You will be doing, like, not only the clinical data managers who is doing PV, who is doing medical coding, everyone in the ecosystem is very, very important and needs to be taken their work very seriously and do ethically, because it has a very larger and bigger impact. So yeah, that is how your data cleaning glimpse looks like and a query resolution looks like. It's just a small example. So I hope, Rishi, you got to understand, and everybody else also. If you still have any more questions, you can definitely ask me. Okay, great. We will just go through a little bit more, then call it a day. Uh, okay, so we will quickly just a moment. Yeah, so we will quickly go through two more slides. So as we are discussing, the EDC system, so I would like to know, I would like to tell you what are the EDC platforms that are getting used in some of the well-known companies. So there are different companies who create all these EDC platforms for us. These are softwares. So we have Oracle Clinical, we have Viva Vault, we have Open Clinica. So like Medidata Rave is a very widely used platform. Like so far I have worked in Medidata Rave from the beginning. So IQVIA's, Pfizer, Novartis, Roche, J&J, so every one of them, they also do. So depending upon the sponsor, it could differ. So there, in a company there could be studies which is operating on Medidata Rave. There could be studies which is operating in Oracle Clinical, Viva Vault, and Open Clinica. There is a lot of upgradation that is happening each and every day with this, this cloud based platforms and help of AI. So there is a lot of modernization that is happening, and new systems are coming on. So by the time you guys will be there and working, so there could be a new platform that you will be working on. So these are in general, this Viva Vault is very new, and then Oracle Clinical, also Open Clinica. Medidata Reeve is there for a very long time, and it is a very robust one. So these are the platforms when you will be going and working. These are the platforms you will be working on. Definitely once you get into the company, they will give you trainings how to operate, definitely. So they will show you there are different trainings that you have to do within Medidata Rave, and also you will get training from the company how you have to navigate, and then how you go to page by page, how you raise query, how you pull reports. Okay, so there are a lot of option that is there are a lot of things that you can do within all these systems. You can pull reports, you can raise query, you can close query, you can see audit trial. So as I explained that you will raise a query, it will go to the site, and then site will update, and then you will close the query. So all these activities that is getting done, there is something called audit trail. So it is like a log. Okay, so it will keep a track of each and every activity. So you have opened the form, demographics form, there will be log where at what time with your initials, it will be lo- logged that you have opened this form at this time, you have raised a query, you have cancelled a query. So this audit trial log. So whenever you are working in a database, you have to be very careful and accurate with what you are doing, but because you cannot afford doing a lot of mistake. And if you are doing a lot of mistake, it can be catchable, because every act of yours is getting logged in audit trail. I will try and show an audit trial report how it looks, and it will show your initial. Suppose I'm Dilruba Haque, so how it will come is Dilruba Haque this time opened at 10:07, opened a query or closed a query with details that this is the query you have closed, you have raised. So all of the activity, this is your accountability. Okay, so you cannot do something in the database and get unnoticed. So everything that you do in your database will be captured in audit trial. So you have to be very careful, very vigilant, and very sure when you are working in the EDC platform. Okay, and next we will discuss is briefly for today, just a moment. Yeah, so we will briefly discuss the roles that is available, and like you will join as a clinical data coordinator, and how you can progress through the life cycle of clinical trial. Okay, so in clinical data management, you can start with a clinical data coordinator, where you do entry. It's an entry level role where you work on the conducts and then closeouts, and handle the query resolutions and other, assist with your other people, and then do some data cleaning activities. For clinical data manage, there next will be clinical data manager, and then next will be senior clinical data manager. Then you become a clinical data management lead, and then project manager. Okay, and then so on and so forth. You can be a lead of a whole project, and then you can further, you know grow and be a full, head of entire. Suppose you are working for a entire responsible for J&J portfolio, like that. So you have enough road to grow on. So this is a, many a times initially when I used to do, it was even more new. And then my friends from my masters, who were doing some research work or something, they used to ask me that if it is a sustainable job platform, that if you can work here for 10 years, then yes, definitely, and you have enough opportunity to grow and grow big and earn a lot of money, and then go to places and then on sites and all these things, depending upon the company structure. Not everybody has the similar structures. So yeah, but then these are the roles you can progress on, and depending upon as and when you increase your level, then your tasks also gets complicated. Initially you do data cleaning, and then you help with your leads, and then as and when you move forward, then you go into study designs, and then more complex activities you do. And then if you are becoming a lead, then you are working with your team members and then managing them, going into the meetings, communicating with the clients, and giving them your study updates. All these kind of activities you have to do as and when you are growing. Okay, so this is an overview of your roadmap basically. So these are the roles you can progress into. And so as I mentioned, so whatever I'm teaching you in this phase, it is very important for your interview purpose, because these are the basics that they want you to know. They don't expect you to know a lot of technical things. They don't expect you to know how to do query management, or how to do a essay reconciliation or other reconciliation, or how to build a ecrf. But they definitely expect you to know what is a protocol, randomization, what are the phases. And definitely from next classes we will start with the regulatory things, the guidelines, ICI and GCP guidelines, 21 CFR part 11 guidelines. What are these, what are their significance? So they definitely expect you to know all these things. So please be vigilant about these things, and make a note of these topics and study about these topics, because these will lead you to the job that you want. And then as and when you go, all this information will come handy, that I will teach you. Mm, I will teach you, and it will help you progress. Okay, but these are the initial things that you will have to learn to get through the interviews. And then in between the week, I will try to take a session wherein we will go through, because I want, as I initially mentioned in the starting class, that you guys are, most of them, most of you are still studying or just pass out or doing something. So you have done your college education, and it is not very interview focused or career focused. So what we want to do, we just don't want to blindly study our course. We have to keep a comparison, that see, this is a job description and this is what you are asked, they are asking, and we will prepare accordingly. So maybe in the midweek, or maybe next class, we will also discuss, see our job responsibility slide, and then discuss that what are the requirements that we are having. And we will take that as our roadmap and work ahead. Okay, so for today, we will conclude our class here. And if you have any questions, you can pop in here. If you have questions further, we always have our group, WhatsApp group, mentor group, where you can pop in your question. Somebody is asking something. Yasmin, ma'am, did you started your career as clinical data coordinator? Yes, Yasmin, I have started right after my masters. I started with IQVIA. That time IQVIA was not IQVIA, it was Quintiles. I started my work as a clinical data coordinator one. And yeah, so IQVIA is a very good company. There, all of them, wherever you are going, wherever you are getting, you should start your journey as soon as possible. So be focused, and always remember that the idea and the goal is to get a job. And so we will be studying, so and we'll be also preparing. I will ask you to simultaneously start preparing your CV. I hope you all of you have a LinkedIn profile, so update your LinkedIn profile, update your details, whatever you're learning, and we will discuss it further, that what all you can update there. And we will do simultaneously, so that it will prepare us. It should not be like we are doing the course and then we don't know where we are heading. Simultaneously we will prepare, so that we can be ready for our what do you say, we can be ready for our job interviews. And similarly, I want you guys to prepare, start working on your CVs, and also start looking, create a Naukri profile, go to LinkedIn jobs, look what are the job descriptions are there, what are the job profiles are there. I have discussed with you guys all the job profiles that is you are studying here in CDM, but not necessarily CDM is the only way you can work, the place you can work. So I have discussed, you go back to the old notes, and then see what are the roles and what they are doing, what they're asking. Look the job responsibility and see, maybe something else is appealing more to you, and you're finding interest in that, and try for all the options. You should always keep your option, you should not restrict yourself, because you should be able to start your journey, be it CDM, be it PV, be it medical writing, medical code. So it could be anything, but you should be it CRA, CRC, anything, but you should start your journey. So you look for job roles, you create your Naukri profile, create your LinkedIn profile, go to LinkedIn jobs, go to Naukri, look what are the jobs available. And freshers, not always they write freshers, so you always have to look 0 to 2 years of experience, and if anything is 0 to 2 years of experience, you are good to apply there. Okay. And simultaneously start working on your CV, send it to me or sir, we will review your CVs, and then we have to start preparing simultaneously, okay, as and when we are going ahead with the course. Okay, so for today we will be resting our case here, and I'll leave you guys to enjoy the rest of your Sunday. Thank you very much for joining in and be there in the class. If you have any question, please let me know, we will discuss. And yeah, you have a great weekend ahead. I think in India it's already afternoon, yeah, 3 o'clock. You have the evening for yourself to enjoy. Thank you very much for all the questions, it was a great discussions. I will try and bring in more samples, or examples or screenshots, where I can actually show you what it looks like in real. Okay, so thank you very much. If you do not have any further question right now, then we will say goodbye for today and see you in next class. Okay, and any question you have, and definitely you have 3 homeworks, you should start working on your Naukri profiles. You should have you work on LinkedIn, and you start working on your CV and finalize your CV, so that whoever is eligible, like the final year students, I believe you guys are having the final year students are approaching final exams, so yeah, so you will be soon eligible. Okay, so yeah, that's it for today. Thank you, Rishi. You Thank you for your wish. Most welcome, most welcome. Thank you so much, ma'am, for your invaluable. You're most welcome, Shantani. I hope you guys can make it, useful for you guys, and definitely, the final goal is. I'm telling you again and again, the final goal is to get a job. So the focus has to be always on that. So work towards it. Okay. Anyways, chalo. We'll wrap up today's class then. Bye.